There you go.
Excellent, thank you. And will you put up the slides?
Yes.
Right. Good afternoon. On behalf of myself and Dr. Siwen Huliskoven, welcome to our SWOG Immunotherapy Committee meeting for the spring of 2021. Thank you all very much for joining this virtual meeting. We have a packed and exciting agenda this afternoon, so we will go ahead and get started. Next slide. So first of all, just an introduction to the SWOG IO committee for those of you who might be joining us for the first time. This is really an IO-centric committee that is disease agnostic. And the goal is really to foster cross-committee collaborations and work on clinical trials and incorporating translational medicine into clinical trials. And for the IO committee, we have the co-chairs, and Siwen Huliskoven and I are co-chairs of this committee. We have a number of committee members who come from various disease committees from SWOG and from the broader SWOG community, and also are working on the development of an IMatch working group, and you'll hear about the Immunomatch study in a few minutes, with the idea really of thinking about some concepts and ideas for clinical trials that can be incorporated in the future into the IMatch clinical trial. Because of the goals of the IO committee, we're really focused. I hear an echo. I'm wondering if people could please mute. Thank you.
Yeah, if you're not speaking, please mute yourself.
Thank you very much. So because of sort of this really, the objectives of this IO committee, which are really centered on incorporating immuno-oncology and translational medicine into clinical trials and being disease agnostic, we work with a lot of other groups as well throughout SWOG, and in collaboration with other IO committees, from some of the other cooperative groups and other groups such as the CMACs, the CITNs, Jackson Labs, Cold Spring Harbor, the SWOG Biorepository, and interface also with SWOG leadership. So really this is a very, a committee that incorporates feedback and has contributions from really all aspects of of SWOG itself, the cooperative groups, and the National Cancer Institute. Next slide. So what have we really been working on so far? I think one of the main things that we have been working on over the past couple of years now, a year and a half at least, has been the development of the Immunomatch clinical trial together with the National Cancer Institute, and including the development of an initial pilot study. And you will hear about that from our next speaker, who I'll introduce in a couple of minutes, Dr. Helen Chen. One of the other things that we've been working on is working with various groups to facilitate IO trial design and inclusion of translational medicine and IO studies across diseases. And this is something that we want to further expand and work on in the future as well. And to facilitate this, one of the things that we have started having are monthly meetings on the first Friday of each month at 2:30 p.m. Central Time. Our next meeting is on May 7th, and I invite members to contact us if they would like to be added to the distribution list, or if they have agenda items and things that they would be interested in bringing forward to the IO committee. In these meetings, we discuss initiatives from the committee, we provide updates on ongoing activities, and investigators present trials and ideas for feedback. So please reach out to us if you are interested in participating in these meetings. So without further ado, I'd like to start the agenda for today's meeting. And it really is my pleasure to introduce our next speaker, who is Dr. Helen Chen. She's the Associate Branch Chief for the Investigational Drug Branch at the National Cancer Institute. And where her work focuses on the clinical development of antibody-based therapeutics, active immunotherapies, MAP kinase and RTK inhibitors, as well as anti-angiogenic agents. And Dr. Chen has been at the NCI with her colleagues really spearheading these efforts for this ImmunoMatch trial. And so it is great to have her present the NCI's vision for this trial and the ideas behind it today. Thank you very much, Dr. Chen, for presenting at today's SWOG Immunotherapy Committee. It really is a pleasure to have you here.
Can you hear me all right?
Clear.
Oh, wonderful. Thank you so much, Sue and Katie, for the opportunity to talk about IMatch. This is really a joint effort with the SWOG investigators, and as well as a lot of investigators in the field and across the NCTN. Can we move to the next slide? I think we have already presented the idea of IMatch several times in this setting. So I'll try to be very brief on the rationale and background for the IMatch, and we'll really mainly focus on what is the update and also about the next steps that you're all very interested in. I want to first acknowledge all the people who have been already involved in this effort. And first and most, the SWOG investigators and the operational office leading the efforts for the overall concept, and also work tirelessly on the pilot trial that we will talk about. And also my colleagues at NCI across many different branches for this very comprehensive, large effort. And also, very importantly, I want to thank the scientific input that we have gotten from subject experts and NCTN investigators in industry. And we have several meetings with these individuals and to really gain the insights in the scientific field and get input for the trial design, and especially for the our first planning workshop back in 2019. And I know Jason is going to speak later. He was involved in that very first workshop also. Next slide. So I think, our idea for IMatch is really to think about a stepwise approach to precision medicine trials for immunotherapy. We know that currently our major challenge in effective clinical development for IO is really the complexity of the system and diversity of resistant mechanisms across patients, and yet we do not have a predicted marker for specific combinations. As a result, many of our immunotherapy trials combinations are done in patients without molecular stratification, and a lot of studies failed in the signal-seeking stage, and they are basically very hard to interpret to understand what was going on. And recognizing the future, true precision medicine will be something in the future, we ask the question whether we can capitalize on our current knowledge and do something regarding prospective molecular profiling, and hopefully that may enhance our the clinical evaluation for our therapeutic strategies. If we think about the IMatch platform, about the assay that had sufficient analytical validation that can be used in clear lab to be used for as an integral marker, there are already PDL1 and as a high commonly used standard of care TMB by next-gen sequencing panel is also relatively well available, at least for some indications. And there's also the gene expression signature for tumor inflammation that also reach the level that can be used in a clear lab. And next slide. So I think some of the markers, this is really just exemplified by the TMB calculation and the tumor inflammation score calculated by the 18-gene panel from NanoString. By combining TMB and TI score, we can divide patients into 4 molecular groups. And these groups with the defined cutoffs can already divide the patients into different probability of responding to checkpoint inhibitors. But more importantly, tumor mutational burden and the inflammation score may reflect the tumor biology, which may have separate patients into different resistance mechanisms that need to have potentially different therapeutic strategies. And if we look at the TCGA data for these each subgroups, we already see they are not evenly distributed within a tumor type, and that probably explains why many of the trials in unselected patients can be misleading or very difficult to interpret. Next slide. So this is the overall IMatch concept. The most important component is this central assay platform. And after a lot of discussion with our experts in the field, it was decided we will include these 3 basic panels. The first is a whole exome sequencing, which will provide TMB calculation and also identify specific variants that may be potentially actionable, and also a panel for platform for gene expression profile to get the score for tumor inflammation signature. And then the IHC for PDL1, which may be usable for some clinical trials. And the platform we want to build in a way such that it is flexible, so it can potentially take new biomarkers in response to a really rapidly evolving field. And with these readouts of the biomarkers, we can divide patients into molecular subgroups based on some predefined cutoffs from current information. And these markers can be used for multiple clinical trials using these markers for either molecular selection or stratification. Very importantly, we do want to build in extensive retrospective analysis, including whole transcriptome sequencing, because these will be very important for us to really understand potential markers for these therapeutic regimens, and it may also inform how the upfront platform should be optimized. Next slide. So it was also decided that before we can launch the IMatch in multiple trials across the NCTN network, we will need to have a pilot study in order to test the feasibility of this platform, whether it can inform patient assignment in real time, and also to obtain some preliminary data on the activity in predefined molecular subgroups. This is mainly because, the IMatch trial is very different from any of the NCI match trial. It will involve, fresh biopsies and also 2 rather complicated assay platforms. So there's technical challenges. But in addition to that, these readouts of the biomarkers are continuous variables. We're going to use our best knowledge to decide what should be the cutoff for our trial. But these numbers may need to be adjusted based on the data from the pilot study and also potentially from ongoing research in the field. Next slide. So the SWOG team had really spent a lot of effort thinking about what can be this pilot study. And they went through various scenarios and for different regimens and clinical settings. And they come down to the concept for this Picasso trial, acronym for biomarker stratified cabozantinib and nivolumab. This is basically a phase 2 trial for the combination cabozantinib and nivolumab in patients with CPI refractory or resistant tumors stratified by tumor biomarkers. And the tumor types to be enrolled are mainly the melanoma and head and neck cancer. And I won't go into details how this setting and treatment regimen is selected, but it's suffice to say that VEGF TKI and the checkpoint combination have demonstrated activity across many tumor types. But in these two specific tumors, the data is still pending. And also the choice of the clinical setting and the trial design should be such that it has to be feasible to complete the enrollment in a reasonable time frame in order to inform the larger IMAGE effort. So basically, as you can see in the schema, patients who progress on PD-1 will all have biopsies and have the IMAGE screening platform, the whole exome sequencing and GEP profiling. And in the stage 1 enrollment up to about 60 patients, they can get treatment before the trial, the assay results come back. Then the feasibility will be analyzed, and if we pass the reasonable, we have a target for turnaround time for the assay between the biopsy and assay. And if we can pass that timeline, then the stage 2 enrollment will start, where patients will have to wait for the molecular results in order to be assigned to a specific subcohort. Now, all patients will receive the same treatment, but we want to ensure that in the end of the trial, we will have 15 patients per histology in each of the molecular subgroups. And the primary endpoint is really to test the feasibility, but also to look at the response rate in the subgroups and to see whether we can, see these response rates may differ between the different molecular groups. And again, the retrospective analysis will be an integrated part of this trial. Next slide. And this is just to show the biomarker for the pilot study. Integral biomarker include whole exome sequencing and gene expression profile. And I just want to add the West report would also, to the patients, will also include tumor or immune-related gene alterations as identified from the whole exome sequencing platform. And the integrated biomarkers retrospectively will have deeper analysis of the whole exome sequencing and RNA-seq and ctDNA and PDL1. And we hope that these retrospective analysis will be important and potentially, can really significantly help us how to think about the upfront platform. Next slide. So what is the next step for IMAGE? So currently the concept is at the CTEP waiting for company review, and the SWOG team has worked really tirelessly on the protocol development. And we hope that it's hard to point to a specific timeline, but we hope this will be sometime this year it can be activated. And however, the full launch of IMAGE, we think, will follow the other two PMI trials. It's mainly because the IT buildup will consume a lot of efforts, and also we want to wait for some scientific results from the IMAGE pilot in order to launch a larger effort. So for this reason, the timeline and formal process for subgroup protocol solicitation is yet to be defined. However, at this time, we really want to continue the engagement with our investigators to gain additional scientific input, not only to support this pilot trial participation from across NCTN, but also to discuss new development in the field that may inform how the central screening protocol might be optimized. We also, should think about how this platform may help enhance the clinical evaluation for the therapeutic strategies of interest. The next slide. So I think thinking about the scientific strategy with this platform, I think, hopefully, and we think we will, the pilot study will establish the feasibility of this comprehensive assay platform that can be used prospectively for patient selection in IO trials. And although currently our biomarker is only based on TMB and TIS scores, potentially as the field is rapidly evolving, the same platform may actually add additional biomarkers, as we already are collecting a list of gene alterations of interest that may be actionable, and there may be signatures for specific resistance mechanism may come up, and new inflammation signatures currently is based on 18 gene, but maybe the RNA-seq platform can be useful. And also, we have a lot of signatures for subgroups of cancer biology with differential response to IO and other agents, which can potentially be all integrated into the platform. And of course, there can be addition of new assays for different biomarkers when appropriate. Next slide. The other aspect to think about scientific opportunity is, to really think potentially from a bottom-up approach. You may be interested in some specific IO strategies or targets, or in a specific clinical setting for a tumor not responding well to immunotherapy, and to really think how this platform may help you address better the scientific questions of interest. I'm thinking about, I was reminded by the Cancer Research Institute landscape analysis. For example, currently only 5% of the clinical IO trials are in the resistance setting. And, that's also because it is a very high bar to overcome resistance once the patient already received the therapy. So there's a very high failure rate in those kind of trials. And we may ask the question whether molecular stratification for potential resistant mechanism may help us to improve trials in that setting. And I know Jason's going to present something about strategies to overcome resistance, so that can be, interesting discussion. Next slide. So this is just my last slide for two components. The first is this essential assay platform, and we're looking for ideas what additional markers can be added. And with the understanding the platform can be used to identify a lot of biomarkers for patient stratification, how you would think about this may enhance your therapeutic evaluation for the areas of interest, could be by the different kinds of clinical settings or different strategies to overcome resistance, and also maybe based on your thoughts, maybe based on specific regimens and therapeutic categories that you are interested. So I think overall, the trial, we're very flexible at this time regarding the trial design, and also the trial overarching structure is also flexible. And I think ideas from really the bottom up will help us to think about what kind of buckets and structure we need to build up for the IMEDGE. So I think I'll stop here, and I look forward to questions and discussion.
Thank you very much, Dr. Chen, for that presentation. We do have, so first of all, I'd like to encourage people to enter questions in the chat, and I will read them out to Dr. Chen. So there is an initial question from Jack Aiello. Are blood cancers included in the IMAT program?
So blood cancer currently is not. We do have cell-lymphoma is not included. We actually have never talked about that. So I guess we can consider. But if we're thinking about AML, there is a MyeloMatch effort also, so it will be, sort of testing that setting.
And then there's another question from Dr. Haider Mahdi. Is the cutoff for the GEP signature, is that going to be the same for any cancer tissue, or an individualized cutoff is going to be used?
Yeah, that's a good question. I think for the current trial in the pilot, we do have some data already from the science paper for the nanostring. So there is a cutoff. They have, tested cutoff, which seems to be the same for several tumors, including melanoma and the head and neck cancer. But moving forward, I think, for cancers we don't have data, then there will be discussion about whether something will need to be generated to make that kind of decision.
And then we have another question from Dr. Kim Margolin. Are the lab correlates being done in the CMAX labs or in individual research labs?
Oh, yeah, I didn't mention that. So this assays will be done for prospective patient selection, so it has to be in a clear environment. So it will be done at the MOCA. We have that. It will not be done at CMAX because they only are for research. So it will be a number of clear labs with we call MDNet assay, including MOCA as the subject.
And then from Dr. Peggy Zuckerman, are there patient advocates who are part of this committee?
This is Dana. Rick Bangs is currently looking for a patient advocate to assign to the Immunotherapeutics Committee. But we have been involving patient advocates in the development of the pilot trial. So Marcia Horn and Sam Guild have both been involved in reviewing the protocol and consent form for the pilot.
Mm-hmm. Thank you, Dana.
Yeah, thank you, Dana. And then we have another question from Dr. Kuderer. As you all know, different cancers have different ways of testing PDL1 by C for drug development. Will we be able to test it in multiple different ways?
Yeah, that's a good question, because initially we think PDL1 may be a marker, across board, but there is a problem with some, we don't know exactly how they perform in all tumor types. So PD1 in our platform, L1 in our platform, may be used for some select trials in the indication where we have some idea how they perform and how to use a cutoff. But it may not be used upfront for all trials, and certainly not, we're not doing this pilot.
Thank you very much, Dr. Chen, for the presentation and for answering all the questions. And thank you, everybody, for asking the questions. If there aren't any additional questions, I'm going to turn it over to Siwen, who will introduce our next speaker.
Thank you, Katie. And okay, there's no more echo. Yeah, and I want to thank Dr. Chen again for presenting the NCI's vision on this very important platform. And I'm really pleased to introduce the next speaker, Dr. Jason Luke. So many of us know Jason really well. He's pretty well known in the tumor immunology and immunotherapy field. So he is currently Associate Professor of Medicine at the University of Pittsburgh and Director of the Cancer Immunotherapeutics Center at UPMC Human Cancer Center. He specialized in early phase drug development for solid tumors, particularly novel immunotherapeutics and biomarkers of immunotherapy activity. And he's a skin cancer melanoma specialist. Jason directs the Translational Immunoinformatics Laboratory, which investigates large-scale informatic approach to advanced cancer immunotherapy. So today he's going to talk about overcoming resistance by targeting the tumor microenvironment.
All right, thanks. Can you guys hear me?
Yes, we can.
Okay, thanks. I think I took control of the screen and then turned everybody's cameras off, and I'm not sure how I did it. So I'm not sure if the tech people can help turn on the cameras back on, or like my video.
I will look.
I apologize. So while that's happening, I just want to say thank you for the invitation to speak today. I think this sounds like a great committee that's emerging, and I think the IMatch protocol is very interesting concept, and it's been in the works for a while, and I'm really excited about to see kind of what comes out of it. While we're trying to figure out the video piece of this, I might just go ahead and start so we don't lose time, although I guess we're a little bit ahead of schedule. In that regard, then I'll say, so again, thank you. So title of the lecture here is Overcoming Resistance to PD-1 Checkpoint Blockade by Targeting or Inducing the T-cell-inflamed Tumor Microenvironment. And many of you are going to be aware of concepts here, but I think to level set, I think this will be a nice talk, and I think it's relevant to the development of IMatch as well. And so in terms of advancing the slides, there we go. So these are disclosures for me in the past year. You can see that I have a lot of interactions with industry, both as a consultant and running a lot of clinical trials. And I keep all my disclosures fresh for viewing on my LinkedIn page if anybody's so of interest to go look. There we go. And so I'm just going to sort of, get start from a position of maybe inviting in people who are maybe new to the field, because obviously anti-PD-1 has become a foundational therapy in cancer treatment. And we're only beginning to learn better how to stratify patients. And on the left-hand side is a cartoon that we published, several years ago now, about the role of PD-1 and PD-L1 interaction in the tumor microenvironment, where we initially thought this was predominantly tumor cells interacting with tumor-infiltrating lymphocytes, and we now know that it's much more complicated than that. On the right-hand side are probably the two strongest biomarkers of anti-PD-1 treatment response that have been identified to date, those being tumor mutational burden as well as T-cell inflamed gene expression profiling. And you can see that in pan-tumor fashion and then across specific cohorts of head and neck and melanoma, that patients whose tumors are enriched for higher TMB or higher T-cell inflamed gene expression are more likely to respond to anti-PD-1 monotherapy. And perhaps in a bit of a paradox that we still don't understand well in the field, it turns out that these two biomarkers actually don't correlate well with each other. And one would think that more mutations would lead to hotter tumors, so to say, but that is not what we see. And in fact, these two biomarkers are not well targeted. And this was a seminal paper published by Cristescu et al. Several years ago now. So if anybody in the audience hasn't read this paper, I would really strongly recommend that you do so. Now, the thing about these biomarkers, however, is that whereas TMB is probably intrinsic to the tumor because the ability to drive high TMB is probably foundational in the development of whatever specific cancer it is, the T-cell inflamed gene expression score is an interferon responsive gene signature that in fact is quite dynamic. So in that regard, then, when we think about targeting a biomarker, this one is a little bit more nuanced. Let's see if we can, here we go. And so the immunobiology of the T-cell inflamed and non-T-cell inflamed tumor is important to understand, especially as we start to think about therapeutics that are in development for advanced cancer. So I was actually junior faculty at the Dana-Farber, and this was the picture actually that caused me to up and move to Chicago to work with Tom Gajewski, who dating back into the sort of 90s and early 2000s was performing vaccine clinical trials where they observed that whereas most patients do not benefit on vaccine clinical trials with cancer, there was a rare subset of patients who would have long-term stable disease and occasionally be responders. And what Tom's lab observed initially from immunohistochemistry was that those were the tumors that were disproportionately infiltrated by CD8 T cells. And you can see that here. And he termed these tumors as T-cell inflamed and did a lot of great work with Stephanie Spranger subsequently to identify that it's really this infiltration of the tumor by CD8 T cells and elaboration of chemokines and cytokines, especially interferon gamma, that leads to the upregulation of the other immune evasion programs that are in mutation-inflamed tumors. And so the most famous of those is PDL1, which you see here, but you can see that others such as indoleamine dioxygenase, as well as the infiltration of FOXP3 positive Tregs are all associated with this T cell inflammation. In contrast, the vast majority of patients with cancer, unfortunately, have never mounted a CD8 response to the tumor, and they never get infiltration, which leads then to the lack of these downstream components. And so this is a really important working model, and it sort of sets the goalposts in terms of thinking about where does a patient's tumor stand on the continuum of T cell inflammation. Tom was also seminal in the field, being one of the first people to bring high-level genomics to cancer immunotherapy bioinformatics. And so he published a paper along with Harlan et al. Dating back some years where they could profile T-cell inflamed tumors in vaccine clinical trials by chemokine gradients. That's outlined in the middle here. And certainly Tony Ribas and many others have contributed to the identification of a true biomarker vis-a-vis treatment response to anti-PD-1 in the context of basically all the antibodies that are in clinical development. And on the bottom here, we see that the progression-free and overall survival of patients with melanoma can be strongly stratified for outcomes by the presence or absence of this interferon gene signature, such that those who have a high signature do well, and those that do not do poorly. And of course, that makes lots of sense and is kind of obvious at this point. But this is important because this is potentially inducible and can change over time. So we'll just go ahead and wait for it here. There we go. So this raises a question for us. If tumors can be phenotyped at baseline as T-cell inflamed or non-T-cell inflamed, is it possible then to develop immuno-oncology approaches for inflamed versus non-inflamed tumors based on this finding? And I would argue that it potentially is, and we have recent data that really suggests that this is the working paradigm, and we'll talk about that in a second. But sort of explore this question dating back a few years ago, we took the tact of sort of generating a non-exhaustive list of IO-relevant drug genes that are relevant for drug development, and correlating those across the TCGA with PDL1 expression, which you can see down here on the right-hand side. And what you can see is that in this heat map, that most of the IO most of the IO-related genes really show up here at the top, being strongly related or strongly associated with PDL1, and that's for the reason that I described before of the T-cell inflamed tumor microenvironment. But more than that, you can see that some of them are more strongly correlated and some are less strongly correlated. So on the top hand, you can see here, are we not seeing the video or the slides?
We're seeing it, Jason. No problem.
Okay,
and we can hear you good too.
Okay, thanks. So what we can see is that some genes are strongly correlated with the T-cell inflamed phenotype, or PDL1 is a surrogate for that, and they're the famous genes you would think. So obviously PDL1 is correlated with PDL1, but CD8 T cells as well, and then a number of other genes, like I said, so IDO and FOXP3, but three that I specifically want to talk about a little bit further here are going to be LAG3, TIGIT, and TIM3. In contrast, there are a number of IO-related molecules that are not so well correlated with PDL1, and to some degree, these would be kind of obvious as well. And so on the bottom, you can see those that don't correlate well, and those would be things like TGF-beta as well as VEGF-A. And again, we know that these are mechanisms of a resistance that mediate immune exclusion and make sense then to not be so well correlated. But this finding that these checkpoint genes are all strongly associated with PDL1 expression is important and has become quite relevant to the field now over the last sort of year or so, with the sort of identification of a couple of combination partners that look to be highly promising. All right. And so just to put this on a little bit more granular level, what we did was analyze the cohort of melanoma patients from the TCGA, stratifying the individual tumors along the continuum of their expression. You can see here of CD274 of PDL1, PDL1 versus HAVCR2, which is TIM3. And what you can see is that essentially where you have high PDL1, you have high TIM3, and it's basically never the case that you would have high PDL1 and low TIM3, and vice versa. It's basically never the case that you would have a low PDL1 and high TIM3. So why does this matter? Well, this matters because it really wouldn't make sense to do a clinical trial where you had low PDL1 and treated with a combination with anti-TIM3, because the target just isn't there. Yet for about 5 to almost 10 years, we've been doing this in IO drug development, where we do expansion cohorts without enriching for PDL1 as a surrogate for interferon gamma. And for that reason, I think we have not really seen the kind of activity that we have been hoping for. Now, there are data now that really suggest that this is what we should be doing. On the right-hand side, once it pops up, our data presented by my colleague Vakar Davar here from UPMC Hillman Cancer Center in Pittsburgh, presenting the combination of a PD-1 plus TIM-3 antibody in non-small cell lung cancer. Now, the thing here was that these patients had previously progressed on anti-PD-1. And you'll see that the response rate, however, or the response waterfall plot here, shows that the responding population in this PD-1 refractory group was really only in those who retained PDL1 after progression. And you can see that this response rate is here. And broadly speaking, this combination hasn't looked particularly active, but I think this is the point, which is that we should be profiling patients even in the resistant setting for PDL1 as a surrogate of interferon gamma, or even better, just to do the gene expression profiling for interferon gamma. So TIM-3 is certainly one example of this. Another you should be cognizant of is LAG-3. And so LAG-3 is a complicated molecule, but it's another secondary checkpoint that regulates T cell function, energy, exhaustion, etc. It has a number of ligands, which we aren't going to get into here, but the Yale group has been instrumental in defining this as FGL1 possibly being one of the most important ligands. But relevant to clinic, what we see is that in refractory melanoma, the molecule relatlimab was investigated in combination with nivolumab. And what you can see is that in a group of patients who had high LAG-3 retained after progression on anti-PD-1, that there was this group of patients, 45% with tumor reduction and about a 20% treatment response. And more than that now, this combination of nivolumab plus relatlimab has gone into a phase 3 clinical trial that's been now reported out in a press release as a positive study. Here we go. The Relativity 047 study. So moving to the front line, where all of you will be aware, patients with melanoma are highly enriched to be PDL1 positive. We see that the combination of a PD-1 plus LAG-3 antibody had a positive endpoint relative to PD-1 monotherapy. And I think this really goes along with the biology, as I was sort of alluding to. We know in this setting that adding CTLA-4 antibody, ipilimumab, to nivo really enriches for response for the PDL1 negative population. And that may be because CTLA-4 has the capacity to prime more T cells and bring more T cells into the tumor. Whereas this LAG-3 combination is really going to be dependent on a tumor microenvironment that's activated by interferon so that you have both PDL1 and LAG-3 expression. The third example I wanted to give here was TIGIT. And again, many of you are going to be aware that I think it was called the Cityscape clinical trial phase 2 study investigating anti-PDL1 with atezolizumab with or without tiragolumab, the anti-TIGIT antibody. It was reported out at AACR now a year ago, showing that in a randomized phase 2 study in PDL1 high patients who have not yet had treatment, there was a benefit to giving this combination. And TIGIT is a pretty complicated molecule for any of you who are following ACR this year. It certainly seems like blocking TIGIT has multiple functions, but one of them appears to be to reinvigorate CD8 T cells. So again, going to our model, the reason that you see activity then for this combination in that PDL1 high population is, again, that's where the interferon signal is. And so that's really where we should be analyzing these secondary checkpoints for combinations with anti-PD-1. And I would say that that's true in the front line. It's also true in the refractory disease setting. Treating patients with a low interferon signal in that setting with checkpoint molecules really just doesn't make sense. That's not where the biology is. We have done some further work to try to look at sort of a more biomarker restricted population to see if there's a way we could enhance patient selection further. We presented this several years ago, no, not several, 3 years ago at ESMO. And what we did was analyze, again, the melanoma cohort of TCGA, and then we did unsupervised hierarchical clustering, but using only those proteins that we know that there are antibodies for in clinic. And so this was a collaboration with Bristol Myers Squibb. So these were drug targets that they had in their pipeline. And what you can see when we perform this analysis is that on the left-hand side, you see the vast majority of the PDL1 high patients, and they have all of these targets lumping together. So the hot tumors is where you see these combinations. And that's probably where you're going to see the most benefit if you're going to treat a total population of patients. What we were interested to observe, however, was that in this sort of intermediately inflamed population over here on the right-hand side, we saw that there appeared to potentially be some biology there that we might be able to target. And so when we look at this group more specifically, you can see that certain groups seem to sort of jump out together a little bit. And so there's a group of tumors here that have high expression of GITR and FOXP3, and we have hypothesized that perhaps those are tumors where the inflamed microenvironment is mediated predominantly by Tregs. We see a group of patients whose tumors are dominated by expression of IDO and LAG3, and we wonder whether or not that could be a population of patients who are having the T cell dysfunction phenotype drive the resistance to anti-PD-1 phenotype. And then we see a third group down here that have high expression of CSF1R and of TIM3, and those are molecules that have also been well described to be expressed on macrophages and monocytes, and suggesting that maybe the myeloid compartment could be mediating exclusion in that setting. So given these data, we've been interested to know, to hypothesize that perhaps even on a patient level, we might be able to analyze the T cell inflamed gene expression microenvironment and then go on and select a therapy based on that. And this is an effort that we've had ongoing for quite a while. The amount of effort that goes into the biomarker development and approval is quite substantial, and I don't have time to get into all of that, but this is a trial that we call ADVISE that I developed with Bristol Myers Squibb a few years ago, and we're now pursuing in collaboration with Dana-Farber, Hopkins, and the University of Chicago, where we take patients with various different PD-1 responsive tumor types, but whom have progressed on an anti-PD-1. We do a treatment biopsy, a diagnostic biopsy, and we send that out for a panel of biomarkers. And based on those results, in a 14-day turnaround time, we can assign them then to a group of different treatment options. And so we've cycled through a couple of different, the ones in gray down here are ones we are no longer pursuing due to a lack of activity, but the ones on the upper top are ones we are still sort of pursuing. And so LAG3, CTLA4, IDO, IL-8, CSF1R, and a number of things. And we're excited about this as potentially a personalized way to apply cancer immunotherapy. So returning to our question then, I sort of talked about hot tumors, so to say, so T cell inflamed tumors, but the same question of if we can phenotype tumors at baseline, can we then choose a therapy based on their immune inflamed status? And so then the corollary question would be, could we do this for cold tumors, right? So again, dating back a few years, our group in Chicago was one of the first to use TCGA to profile, broadly speaking, the T cell inflamed status across tumor types. This is now quite old hat, but when we did it back in 2014, it was kind of cool. And what you can see on the bottom is a splay of individual tumor types with their relative T cell inflamed tumor microenvironment. So you can see an increasing frequency of T cell inflamed samples as we go up kind of to the right-hand side. And we found this to be a lot of interest because this was before the approval of anti-PD-1 in most of these tumor types. What you can see is that those that tended to be responsive tended to be mostly over here on the right with some outliers, lung, kidney, melanoma, head and neck, etc. But this said that we were probably onto something with this sort of profiling procedure. Now, one thing that we found that was of a lot of interest was that when we looked at the non-synonymous mutational load, as I sort of described at the beginning here, we saw even way back then that there was no correlation between the number of non-synonymous mutations and our T cell signature, so that the median was no different between hot and cold tumors in terms of mutations. We took this even one step further, which was we selected candidate HLA-A2 binding peptides and synthesized them, and then still showed that even those binding peptides were actually no different between hot and cold tumors. And this was important because back then, and maybe even still now, one of the open questions was, well, in tumors that lack a T cell inflamed phenotype, are there antigens that you could actually target? Because if you have to have a high mutational burden in order to have a hot tumor, then in low mutational burden tumors, then there would be no possibility for success. And we saw that was not the case, but rather the presence or absence of the inflamed status had a lot more to do with antigen presentation. And so you can see down here that BATF3 gene expression, and BATF3 being an important transcription factor in dendritic cells as well as T cells, was associated with T cell inflamed tumor microenvironments, as well as other elements of the interferon response machinery like IRF8. So this really told us that if you could do it properly, you might be able to generate inflammation in non-T cell inflamed tumors that you might be able to then harness with checkpoint blockade. And I'm going to show a few examples of how we have done that so far. So one of these was to combine checkpoint blockade with another therapy. And so at ASCO last year, we presented this data for combining margetuximab, which is an Fc-engineered anti-HER2 antibody with doublet checkpoint blockade. And I show this here because the Fc engineering matters here. So by binding to HER2, this molecule margetuximab can then attract NK cells via the enhanced ADCC functionality of the Fc engineering in this antibody. And what you see in the middle panel there are peripheral blood mononuclear cells exposed to gastric cancer cells, and then the antibody on top of that. And you can see that in the context of control, you have little presence of these various different cell populations, but when you add in the margetuximab, you actually get an induction both of innate immunity with the NK cells and the monocytes, but you also get an induction of adaptive immunity with the CD4 and CD8 T cells coming in. So this is exciting because if we're getting T cells in the tumor microenvironment, then we're probably getting interferon, and we might be able to block checkpoints. And so we did that in the combination of margetuximab with tebotelimab, or MGD013. And MGD013 is a bispecific DART molecule that binds both PD-1 and LAG-3. And what that led to preclinically was an enhancement in these cell culture models for ADCC and NK cell killing. So you can see the numbers of live cells here with control or MGD013 alone, and you see no real difference. But if you add in the margetuximab, you now all of a sudden get a major increase in the amount of cell kill. So translating that into the clinic, then we performed a phase 1 trial where we combined margetuximab plus tebotelimab. And I won't go over all of the data on the slide here just for the sake of time, but suffice it to say that in a population of HER2 refractory tumor types, which at baseline were found to have very low levels of PDL1 and LAG-3, when we combined this treatment of margetuximab plus tebotelimab, we got about a 40% treatment response rate. And this treatment was very well tolerated. And I think that this is an exciting sort of example of where we might be able to go combining putative standard treatments in air quotes and enhancing actually the immunological effects of those therapies, perhaps to overcome resistance and to facilitate checkpoint blockade. Another example of this would be the question of after progression of anti-PD-1, should we continue anti-PD-1 or give ipilimumab alone? So in melanoma, all of you would be aware that standard of care was ipilimumab until the KEYNOTE-006 study, which established pembrolizumab as a frontline standard of care. And an interesting aspect of that clinical trial was that patients who had progressed on pembrolizumab, just as part of standard treatment, went on then to get ipilimumab monotherapy. And the benchmark from this population of these 97 patients who did this was about a 13% overall response rate. And there are a number of other papers that also confirmed this, suggesting that there was a modest but real activity level for ipilimumab as a monotherapy after anti-PD-1 of about 13%. There was a rationale, however, to suggest that if you are bringing in new T cells with ipilimumab, perhaps some of that activity is getting blunted without the continuation of anti-PD-1. And so based on that hypothesis, I initially launched an investigator-initiated clinical trial, which my fellow Daniel Olson, who's now junior faculty at the University of Chicago, carried forward to perform a study where we rolled patients over. Those patients who progressed on anti-PD-1 in the front line went on to get pembrolizumab plus ipilimumab, and you can leave it aside. We used a lower dose of ipilimumab, but it's not really the point here right now. And so these were the results of that clinical trial, which we presented at ASCO a couple times, and I actually got the page proofs from JCO today, so I'm excited this will be published very soon. What you can see is a substantially greater response rate to continuing anti-PD-1 and adding IPI on top of it, at least in melanoma. And so we saw a response rate of about 8-ish%, and you can see that some of these responses are quite deep, these including complete responses, and that the median duration of response on this clinical trial was about 18 months. So these are real IO-like responses, even in the second line. And what was really interesting, relevant to this talk, was that when we profiled the patient's tumors at baseline in the study, we saw that the vast majority of them were either non-T-cell inflamed or intermediate, which would make sense because these were the patients who progressed on anti-PD-1, but that this is where the responding population was as well. And so the vast number of the responders, so the orange and the red PRs and CRs, were in this group, again, suggesting that just because there's an initial progression on PD-1 and a baseline non-inflamed tumor, it's possible that combinations could overcome that resistance phenotype. One third story here, just quickly, that we presented at AACR just a little bit ago, a couple days ago actually now, was the combination of anti-PD-1 plus a TLR9 agonist in PD-1 refractory melanoma. And so on the left-hand side, you can see a drug called vidutolimod, which was previously described as CMP-001. It's a very interesting drug. It has two component parts. One is a CpGA DNA payload, which is a G10 TLR9 agonist that induces type 1 interferon. And that's encapsulated in a virus-like particle that makes it more immunogenic. The virus-like particle induces Q-beta antibody formation, and that is then facilitates uptake into myeloid cells, particularly plasmacytoid dendritic cells. And the results of this study had previously been presented, but I'll just highlight here that even in PD-1 refractory tumors, there was a 23.5% response rate, and its treatment was generally speaking well tolerated. The point of our presentation at ACR, however, was to look at baseline correlates of treatment response. And just very briefly, you can see that across the cohorts of interferon gamma gene signature and across non-synonymous mutational load, that the responders, which is in the right-hand column for both, so CRPR and IPR, did not have a statistically significant difference for this combination of PD-1 plus a TLR9 agonist. Again, suggesting that the TLR9 agonist might facilitate the induction of an inflamed microenvironment even in tumors where this had not been present previously. And what these data on the upper left show was that after re-exposure to the drug over time, there was an induction in the peripheral blood of CXCL10 or IP10, and that this correlated strongly with the induction of treatment response, again suggesting that you could activate the tumor microenvironment with the TLR9 agonist. And the bottom on the right just showed that in clinical responders, we saw an induction just as we would expect of type 1 interferons, shown here, as well as interferon gamma gene signature or adaptive type 2 interferon, as well as the influx of CD8 T cells. So these three examples, I think, show that even in patients who at baseline don't have perfect IO biomarkers, it is possible still via combination approaches to overcome that non-T-cell inflamed tumor microenvironment. The final quick story I want to tell surrounds other molecular mediators of T-cell inflamed and non-inflamed tumor microenvironments. And I don't have time to go through all of this except to say that we, working with Tom Gajewski and then having carried this forward to Pittsburgh, are studying the impact of various different parameters of inflammation at the level of somatic differences in tumor cells, meaning which mutations are different between tumors, the germline genetic differences of the host, polymorphisms in immunoregulatory genes, as well as environmental differences such as the commensal microbiota, which I don't have the time to get into. But one story I was going to just tell very quickly is some of the work we've been doing around distinct oncogene pathways being activated in the tumors of different patients. And so Tom's lab with Stephanie Springer published this very famous paper now in Nature in 2015. And to summarize it very quickly, some tumors, about half of non-inflamed tumors in melanoma, activate the Wnt beta-catenin signaling pathway. And active Wnt beta-catenin signaling actually suppresses chemokine gradients such as CCL4. And that leads to lack of bad F3 dendritic cells sampling the tumor, and therefore failure of T-cell priming and lack of response to checkpoint blockade. And they showed this very nicely in GEM model of BRAF P10, where you can see that at the baseline, these tumors are responsive to checkpoint blockade, but that if you engineer in the beta-catenin transgene, you lose that responsiveness. So building on their work, we expanded this model of beta-catenin mediated immune exclusion across basically all tumor types of the TCGA. And you can see on the left-hand side a couple of examples where high levels of beta-catenin protein almost universally correlate with lower levels of T-cell inflamed gene expression. Or another way to say it is beta-catenin drives immune exclusion. And this is true across these examples shown here. And we were able to categorize tumors across different tumor types showing here. And not very surprisingly, tumors that are generally speaking IO resistant, such as cholangio, ovarian, HCC, are those where we see high levels of beta-catenin. And if we had good beta-catenin inhibitors, might be a strong priority for consideration of IO combinations in those tumor types. We expand this model beyond beta-catenin and published this last year in Genome Medicine, where we did a number of different layers of analysis, and I don't have time to go through all of them, but just a couple of examples quickly was that in a per tumor type analysis, we looked at all of the recurrent non-synonymous mutations and their association with the T-cell inflamed phenotype, and a few example tumor types are shown on the upper left-hand side here. And I'll focus on a few things because we did this work actually quite a while ago in about 2015, but only published it last year. And we were excited to see that a number of these in silico observations actually have held up in clinical trials, which is that we observed that mutations that KEAP1 disproportionately associate with a lower response rate to checkpoint blockade in lung cancer. and that mutations in PBRM1, the epigenetic regulator in renal cell cancer, actually associate with a stronger outcome or a better outcome to anti-PD-1. We also took the approach of trying to look at the T-cell inflamed tumor microenvironment and recurrent mutations across the entire genome, and looking for recurrent mutations that we think may mediate immune exclusion independent of tumor type. And you can see when you look at this list, it probably looks overwhelming. And obviously, I'm going kind of fast, so you can look at this paper if you find it to be of interest. We were interested to see that beta-catenin, or CTNNB1, was actually our second strongest hit in our pan-cancer analysis. But very interestingly, the highest hit was actually isocitrate dehydrogenase 1, IDH1. And all of you are going to be aware that that's a highly expressed, or a gene that's commonly mutated in gliomas. But it turns out there's a pretty substantial fraction of patients who have these mutations in other tumor types, such as cholangiocarcinomas and sarcomas and GI tumors, et cetera. And this is all great work done by my colleague and longtime collaborator, Rir Bao, who's my co-PI for our lab. And so why are we interested in IDH1? Well, about that same time, one of our colleagues here at Pitt was doing his postdoc and published this paper in JCI looking at the impact of IDH inhibition on vaccination in a glioma model. And what I'll focus your attention on is down here in the IDH mutant population. What you can see is that if you try to vaccinate these tumors, they get some level of benefit relative to vehicle. But if you add the IDH inhibitor, which is this black line here, you extend the survival of these animals quite substantially. And this effect was actually associated exactly with the biology that we're looking for, which is the induction of CD3, CD8 T cells in the tumor microenvironment. And it actually happens both in the context of the wild type mice, as well as the IDH mutant mice, which is of interest, suggesting it's possible that IDH inhibition may have a more active role beyond just the mutation. Here on the right-hand side, you can see that similarly, this was associated with induction of interferon gamma, which would link to the T-cell inflamed tumor microenvironment. So based on these data, we've launched a phase 2 clinical trial combining ivosidenib, which is the IDH1 inhibitor that's approved for hematologic malignancies, with nivolumab in a genomically selected population of patients with advanced solid tumors who have IDH1 mutations. And this is a highly translational trial with a collaboration between our lab as well as Greg Delgoff's lab here at UPMC Home and Cancer Center. We're going to be looking at changes in interferon responses, epigenetic and metabolomic changes surrounding the TCA cycle, as well as peripheral blood, immune, and ctDNA monitoring. Funding is coming from DoD, Agios, and Bristol Myers Squibb, and this is a collaborative effort with one of my best buds at Sloan Kettering, Jim Harding, who's in the GI oncology service there. So in conclusion, I guess I would say the combination immunotherapy is already a current and near-term future in several different cancer types. And the T-cell inflamed tumor microenvironment really may serve as a model for predicting rational combination immunotherapies. Targeting of mutations or pathways associated with immune exclusion may present rational immunotherapy combination approaches to overcome the non-T-cell inflamed tumor microenvironment or to augment immunotherapy in T-cell inflamed tumors. And finally, multiple clinical trials preparing for activation or in development, and I think that's obviously the point of this committee. And so I really look forward to the ideas that would come out of this committee really to enhance the field and move it forward. And finally, I'd just like to acknowledge my funding sources, people in my lab, as well as our collaborators. You can check out our Twitter handle in our lab if you are so interested. And on the right-hand side is Checkpoints Band, of which I am a modest member playing the trumpet. And I would be very excited to say that we're still hoping that SITC 2021 will be live. And if so, please come to the concert. It'll be super fun. So with that, thanks very much. I'm happy to answer any questions.
Thank you so much, Dr. Luke, for such a great presentation. So I encourage our audience to post your questions to the chat area, and I can help to ask the questions. While we're collecting those questions, so I have a question, Dr. Luke. So it's really the heterogeneity of resistant mechanisms that's really in the way of developing effective combination strategies, right, to overcome resistance to immunotherapy, specifically PD-1 checkpoint inhibitor resistance. So, and there are many efforts to try to develop biomarkers for each different combination. It has not been overly successful. And therefore, on one end, we have a lot of promising combination treatment for our patients. And, as a frontline, a clinical trial, a person who develop and execute clinical trial, we all know, these kind of combination can really help patients. But on the other hand, we don't have a lot of positive, late phase clinical trials to get these combination into market. So, there are a lot of biomarkers that have been developed and nicely outlined in your talk to help the further development of these combinations. But it's really, it's not very clear how best to utilize these combinations or the biomarkers. So I'm just curious, what do you think the next step should be for the field in the testing of combination strategies? You outlined a BMS trial where you actually use the biomarker, to try to understand the patient's tumor and the tumor microenvironment and potential mechanism of resistance and design possible combination that might work. It's a little different from the IMatch approach. In that setting, in IMatch, we use biomarkers to try to separate patient into different biological groups, but all the patients will get the same treatment, so we know which biological group is going to be active.
Yes, I didn't really have time to get into it, but it's like several layers of question there. They're all good ones. I actually, maybe it wasn't clear enough. Maybe I was too nice about it. I actually think it's quite clear what needs to happen, which is that people need to have a hypothesis around how the drug works, and then they need to actually test that hypothesis. And I would actually argue that for at least the last 5, maybe 10 years, we haven't been doing that. So the example of TIGIT and LAG3 being active, but only in the population where you enrich for the interferon response, goes exactly along with the biology. So to me, there's the answer. So if we think it's an interferon-inducible gene that we're targeting, then you have to do it where there's interferon. So then you have to enrich the population for high PDL1 or interferon gene expression, et cetera. And so when we think about how many, how many different combos have been developed, we really haven't done that in terms of selection in expansion cohorts on phase 1 trials. And so I would argue that if we're developing new molecules, we need to have a hypothesis, and we need to make sure that at least a certain percentage of the population that participates actually has that biology. Otherwise, we don't really know whether or not we're testing the question. So, I would argue that in expansion cohorts, to say there's a new novel checkpoint, the patient should have biopsies, and as an integral evaluation, we should know what their PDL1 status is, potentially whatever the marker it is that we're going after in real time as we're putting them on the trial, so that if we get halfway through the expansion cohort and we say it's like PVRIG or something, if none of the patients have expression of PVRIG and we're trying to block it, well, then we haven't really tested the hypothesis. And we need more of that. And I think the hard part is that runs up against timelines for clinical trial accrual, because you're going to end up having to treat more patients than you wanted to in the clinical development. And a lot of drug companies just straight up don't want to do that. They just want to get the patients on, and they kind of just think, well, the drug will either work or it won't work. And so I think we need to be more read into that further, because I think there certainly are combinations that could work in an interferon high population that aren't going to work if you do it otherwise. And then when it comes to other biomarkers, I think, getting more specific, I think that's a real good point. In our trial, we're trying to piece out for an individual patient, does it matter if they're LAG3 high or, CTLA4 high or something like that? I think that remains to be determined, but I think there's at least enough information for us to chase that fundamental biology around the interferon response.
Yeah, absolutely.
You just went on mute, Siwen.
Sorry. I just agree with you. Biomarker is the key. What's your advice, Dr. Luke, for the drug developers when they just start looking for biomarkers? It's kind of difficult, right? Because there are so many different resistant mechanisms.
Yeah.
What would be your advice?
Well, what I would say is that we should build off of what we already know. And to that point, then, and I've kind of actually argued. I know Dr. Paletti is from Yale. I've already argued with Mario about this. Mario Schnoll sometimes takes the tack that we just don't know enough, but I feel like we know enough that we should be profiling every patient who's on an IO clinical trial by their TMB and interferon response, and that should be like the baseline. And then if you want to develop on another biomarker, you have to know where that biomarker sits vis-a-vis those other two that we already know. So to me, that would help us a lot, because if it's true that your second biomarker tracks really tightly with PDL1, which seems to be the case with TIGIT, and it seems to be the case with LAG3, well, then it's not really going to be that much of a different biomarker, right? So we need to like build off what we know already and then incorporate whatever the new biomarker is going to be. And I think IMatch actually does a great job of doing that.
Yeah, excellent. So Katie has a question. Katie, I don't know whether you want to ask directly or you want me to read your question.
Sure, I'd be happy to ask it directly. Dr. Luke, thank you very much for your great talk. One of the questions that I had was related to the somatic mutations and how that are associated with either response or lack of response to immunotherapies. And I was just wondering how we should be thinking about that in the context of clinical trial design and sort of how to best approach it. I also noticed that some of the genes that are involved are different in different cancers. And so how should we as a community be thinking about this?
Yeah, I think that's a really good question. That's actually a hard one. I don't know that we know enough yet to really drill into that to make it a differential biomarker. So I made the example of KEAP1, but clearly it's not the case that like if you have a KEAP1 mutation in lung cancer, you never respond. It's just the response rate is lower. So in terms of the specific mutations, I think it's a little bit harder to try to use that as integral biomarkers. We picked IDH1 because that's the one that has the cleanest phenotype. I mean, almost everybody who has an IDH1 mutant tumor has a cold tumor, so to say. But I think it's definitely much more complicated than within tumor types trying to think about that. So I don't have a great answer about that other than to continue to follow it. And then someday, I think, the future someday is going to be sort of multi-modular biomarker plans where you can say a little bit of this and a little bit of that, and what do we think the best treatment's going to be. At the current time, I think we're mostly still in sort of data gathering mode in that regard.
And I think, one of the interesting things is, for example, when you brought up KEAP1 in lung cancer, we know that that's also associated with a decreased sensitivity to other therapies, targeted therapies and chemotherapies in lung cancer. And so I think it's going to be really interesting to look and see which are the somatic mutations in tumors that are just generally associated with decreased sensitivity to various different cancer therapies, and then which ones instead are specific for immunotherapies versus other therapies. So I think that's really another area that we have a lot to learn about still, but one to keep an eye out for.
Yeah, well, I just, we're all immunotherapists here, so everything is immunotherapy, right?
Right.
All right. So thanks again, Dr. Luke, for such a great presentation. And I will turn the podium to Katie again to introduce our next speaker.
Thank you very much, Siwen. So it's my pleasure to introduce our next speaker, Dr. Sar Gill from the University of Pennsylvania. Dr. Gill is an assistant professor of medicine at the University of Pennsylvania and scientific co-director of the cell therapy and transplant program there. And today he's going to tell us about chimeric antigen receptor macrophages for cancer treatment. Thank you very much, Dr. Gill, for joining us today and talking about your work.
Thank you for the invitation, Dr. Paletti and colleagues. Really a pleasure to be here and to listen to some of the things that are going on in solid tumor immuno-oncology. Let me see if I can take control of the presentation. I know that I was given it. Looks like that is working, I think. Okay, so most of my work has been in the hematologic malignancy space, but the concept of CAR macrophages is quite new and is squarely aimed at solid tumors. And so I'll take you through it shortly. Firstly, I have some disclosures, the most relevant of which is that I co-founded Charisma Therapeutics, and that is a company that has now brought CAR macrophages to the clinic. I'm not in the company, though I do own a tiny, tiny part of it. So where the story starts off is the landscape of CAR T cells for liquid and solid malignancies. This is a really nice paper published last year from a group at Cornell and Memorial Sloan Kettering, which, although very busy, this sort of pseudo heat map represents the hotter colors are the number of clinical trials up to greater than 100, and the white, of course, represents no clinical trials in any given space. Liquid on the left, solid malignancies on the right. Various indications are listed vertically, and various potential cell surface targets, which of course are how you target CAR T cells to cancer, are listed on the horizontal there. And so you can see the relatively small number of hematologic malignancies, but colored with some pretty hot colors, meaning a lot of CAR T cell trials done in that space. In contrast, you can see a pretty sparse and white map over on the right in solid tumors, with some exceptions, with some small number of trials in pancreatic, for example, mesothelin, etc. So the reason for that, I think, is that there have been pretty low response rates. For example, listed here, you can see various targets: CEA, HER2, GD2, mesothelin, and EGFR variant 3. Really just a small number of trials using CAR T cells against those antigens. And the response rates are really quite low. Of course, the ones that are higher are GD2 in kids with neuroblastoma. So the challenges that are posed by solid tumors for adoptive cell therapy in some ways are probably similar to the challenges that you heard about from the previous speaker when it comes to recruiting tumor-resident T cells. And I think of them as in three buckets. The first one is trafficking and penetration. That's not necessarily something you hear a lot about, but in the adoptive cell therapy field is something that we do think about. And so T cells require signals to home and then extravasate and penetrate the tumor microenvironment. Then they need to survive and thrive within the tumor microenvironment, and that relates to some factors that you heard about previously, including active immunosuppression by the tumor cells and their surrounding immunosuppressive cells, exhaustion, metabolic challenges that are posed by the tumor, poor proliferation and persistence within the microenvironment. You heard a little bit about, or actually quite a lot about, antigen presentation, and then the cytokine milieu that is favorable or otherwise to T cell activation. And then something that is, of course, crucial for any type of T cell immunotherapy is what is the target, and is that present homogeneously on all the cancer cells, or is it heterogeneous? And generally speaking, for both intracellular targets, which are ones that of course, to which T cells have access through their T cell receptor, or membrane targets to which T cells and antibodies have access through basically antibody-mediated like recognition. In solid tumors, those are generally heterogeneous, and that relates to all targets that we've thought of, with a possible exception of EGFR variant 3, which seems to be largely specific to cancer, although, for example, in glioblastoma is really not homogeneous. It may be specific to the cancer, but is really not homogeneous across all cancer cells within a given patient. So we, my grad student and I, quite a while ago now, sat back and thought about these challenges, and I suppose from the prism predominantly of the trafficking and survival within the microenvironment. Otherwise stated, the question could be, what immune effector cells or potential immune effector cells actually do appear to be well suited to survive within the solid tumor microenvironment. And so you can ask it in a variety of ways, a variety of different databases, and generally speaking, what you come out with is that the most abundant immune cells within solid tumors are macrophages, various different flavors, other myeloid cells as well, whereas T cells not necessarily. And of course, we think these days about a T cell inflamed or not inflamed tumors, as you heard about very recently in great detail. And so why are myeloid cells abundant within solid tumors? This is, again, a busy slide that we don't necessarily need to go through every single aspect thereof, but I think essentially it comes down to their ability to invade or intravasate into the tumor. And some examples include chemokines, but also cathepsins and matrix metalloproteinases that allow the cells to penetrate, resistance to therapy, and angiogenesis. So what I mean by that is that if I can sort of anthropomorphize a tumor for a second, if you were a tumor, if you were a metastatic cancer, what do you want around you? What immune cells would you want around you? Well, you want ones that will facilitate metastasis, invasion, resistance to therapy, maintenance, immune regulation, inflammation, angiogenesis, and generally speaking, immunosuppression. And so we think of tumor-associated macrophages as the bad guys, but that doesn't necessarily need to be the case. I mentioned chemokines. We've done some profiling, and I'll introduce the concept of CAR macrophages in more detail in a second, but we've done some profiling on so-called M1 or M2 or our engineered CAR macrophages, showing in this heat map with the deeper blue colors, higher expression of a variety of different chemokine receptors. And in a xenograft mouse model, which is the predominant workhorse as such in my lab, we were able to show that adoptively transferred macrophages, when injected intravenously, these macrophages were labeled first, and when injected intravenously, home largely to the tumor. You can see here tumor, lung, spleen, and liver dissected out of a mouse. And the brightest colors, of course, represent the greatest numbers of labeled macrophages that traffic to those tumors. And so that in this one slide, I show you how and why, and I show you the observation as well. So macrophage therapeutics are not new. You could even say that many different monoclonal antibodies are to some extent macrophage-based therapeutics, since they recruit antibody-dependent cellular phagocytosis and maybe ADCC a little bit as well. Newer modalities, of course, are CD47 blockade, so blocking the inhibitory CD47 SIRP-alpha do not eat me interaction, and which I think of as a macrophage checkpoint, immune checkpoint. There have been some attempts to block monocyte recruitment, if you think of monocyte macrophages as the bad guys in the tumor. So blocking CCR2, blocking CSF1 receptor in order to diminish the ability of tumors to recruit into themselves those suppressive cells. The chief hurdle there appears to be that as soon as you stop doing that, as soon as you stop administering the drug, there is just this rebound of chemokine signaling, and then those immunosuppressive cells get drawn into the microenvironment. So when we started designing this therapeutic, we took a page out of the engineered T cell therapy field. Those, of course, allow us the potential to exert multiple effector functions, T cells do, but so do macrophages. And so if we think of macrophages initially as being designed or evolved, I suppose, to phagocytose senescent or infected cells, we could potentially imagine that they could also specifically be made to uptake and kill tumor cells too. So the schema shows you an overview of what we found, and I'll show you in the next few slides the primary data as well. We made macrophages that bear a chimeric antigen receptor, pretty similar but not identical to the one that we put on T cells. They, through the specificity of that CAR, recognize tumor-associated antigens on cancer cells, phagocytose them, are induced to be activated and produce a variety of effector cytokines and chemokines, and are also shown to upon phagocytosis to present tumor-associated antigens, and thereby function both in a specific direct anti-tumor mechanism, as well as an indirect one that presents antigens, stimulates T cells through their T cell receptor, and thereby a vaccine-like effect as well. And the overview of this platform is that we spent quite a lot of time developing an approach that would successfully genetically engineer human macrophages. This is actually really quite difficult, and that the virology tools that one uses for T cells don't work very well at all on macrophages that appear to be resistant to, for example, lentiviral or even AAV-based transduction. And so eventually we came across this chimeric adenoviral vector called Ad5F35 that actually is exceptionally good at transducing macrophages, human macrophages, to a very, very high level. And in addition to that, and this was initially, I would say, an unexpected and sort of unthought of potential benefit, but one that actually has become quite apparently very important, is that exposure to the virus, the viral vector itself, being an adenovirus that does not kill the macrophages, but exposure to that virus mediates a very strong M1-like polarization, if you like. And I'll show you some data on that, and that's also in our paper. So basically, you get a 2-for-1 deal. The very vector that gives us this exceptionally high expression of the chimeric antigen receptor, much higher than you would get with any other vector that we tested, the same approach also gives you a very strong M1 polarization with the production of a variety of cytokines and enzymes like the ones I mentioned before, cathepsins and matrix metalloproteinases, chemokines, hormones, peptides, antigen presentation, and that was the platform that we sort of came across. One of the questions that we were banding around initially was, is this just a fancy way of putting an antibody onto a macrophage? After all, macrophages can bind monoclonal antibodies through their Fc receptor and thereby be shown to exert a specific effect. Well, the answer is no. Here are 2 different experiments where we exposed our anti-HER2 redirected macrophages to a breast cancer cell line and compared that to the use of trastuzumab Herceptin with control macrophages. And so you can see, for example, on the left that the targets alone, no phagocytosis, macrophages alone, no phagocytosis, macrophages with Herceptin, a little bit of phagocytosis, but the HER2 CAR macrophages, which, by the way, the HER2 CAR, the scFv is identical to the one from Herceptin, exert much higher phagocytosis, and the same data you can look over to the right can also be shown by inhibition or in fact rejection of the tumor. This was a different experiment where we also tested pertuzumab as well. So as I mentioned, a lot of the initial work, at least, was done using xenografted mice. That's just generally what my lab does, but I'll show you in a moment some immunocompetent mouse experiments, which are very important too. Here we showed that we can significantly prolong the survival of immunodeficient mice that were engrafted with a HER2 overexpressing cell line and then treated with control macrophages in red or with CAR macrophages in green, significant excessive prolongation of survival there. We studied in great detail the effect of the macrophages that we engineered to become CAR macrophages. A key question for us was if those macrophages come into the tumor microenvironment and come into proximity to the pre-existing M2 type or tumor-associated macrophages, whatever you want to call them, which direction will the interaction result in? So will the pre-existing tumor-associated macrophages polarize our M1 CAR macrophages to become immunosuppressive, or will the opposite occur? And every time we looked at this, what we found was that the effect of CAR macrophages was a dominant positive effect, if you like, on the surrounding M2 macrophages. And you can see some data here, very simple. Initially with flow cytometry on the left, you can see we polarized macrophages to M2A or M2C, which is a sort of essentially fancy way of and very artificial way of polarizing macrophages with a variety of different cytokines. And that exposure to the CAR macrophages led to the upregulation of these activation markers. And then using RNA sequencing, and I'm showing you just a few of the pathways that were enriched there, you can see that the original TAMS M2 immunosuppressive macrophages now become highly activated by virtue of their exposure to the M1 the CAR macrophages. In addition, these CAR macrophages can recruit and prime T cells. So on the left, you can see we quantified chemotaxis, either exposure to resting T cells, exposure of resting T cells on the left or of activated T cells on the right to media, that is to say secreted products from control, UTD or untransduced macrophages in blue, or to CAR macrophages in red, showing that whether you were a resting T cell or an activated T cell, your chemotaxis was increased upon exposure to CAR macrophages. And then we developed a fairly complex but ultimately very specific assay to show that these CAR macrophages can present antigen and stimulate antigen-specific T cell proliferation. So the readout on the right is proliferating T cells upon exposure to media, control macrophages, CAR macrophages alone, antigen-loaded CAR macrophages, and the antigen here is NY-ESO, or antigen-loaded CAR macrophages, again with NY-ESO. And so the NY-ESO-specific T cells proliferated most highly in response to the CAR macrophages. And so that was all with xenografted mice, and that most of that was published already in our paper a year ago. But we then went to do some work with immunocompetent mice in order to study some important questions about combination therapies, as well as the potential for a vaccine-like effect. So here we used a CD26 HER2 colorectal cancer model, which overexpresses HER2, and this was made to overexpress HER2 in this instance. You can see that in the top right of the slide, the tumor volume or tumor growth over time upon exposure to untransduced macrophages or untreated at all, no difference there. But upon exposure to CAR macrophages, you can see that the tumor prolonged control of tumor growth in some of the mice, not all of them, and that translates to a survival advantage there, as you can see in the bottom left, in the bottom right as well. Now, the preceding in vitro data that I showed you in a few macrophages to recruit T cells to the solid tumors, I guess you can think of that as making cold tumors hot, perhaps. So control macrophages on the left, CAR macrophages on the right were injected into these CT26 HER2 overexpressing mice, and then you can see that when we look at the tumor microenvironment and stain for anti-murin CD3, you can see two things really. The clearest one, the most obvious one, is that we have much more infiltration of T cells into the CAR macrophages treated mice. And in addition, you can see that really the inset, the enlargement there, you can see that those T cells are bigger and more activated as well. So that suggested that perhaps we do, in fact, have the ability to induce a vaccine-like effect. And so we designed the following experiment, whereby, again, immunocompetent biopsy mice bearing the CD26 tumor overexpressing HER2 were treated with CAR macrophages, and the complete responders were then re-challenged a few months later with the parental tumor, the CT26, but one that was not overexpressing the HER2. And this would sort of model, I guess, an antigen negative escape or immuno editing, if you like. And the controls were naive biopsy mice that were challenged with the CD26 wild type. And what you can see on the top right is that tumor grew in the naive mice, but tumor did not grow in the previously, 2 months earlier, treated CAR macrophage treated mice, suggesting that we did indeed induce immunosurveillance, essentially, one of the holy grails of immunotherapy, and that was carried out to the last follow-up. So so that was preclinical data. As I mentioned at the start, and just to be super clear, I then co-founded this company with my erstwhile grad student, and I now know I'm certainly not in the company, nor am I suggesting that anything about the clinical trial, but I think it is of interest and probably one of the reasons I was invited to speak to you today is that there is now a therapeutic in the clinic in a phase 1 clinical trial at Penn and at the University of North Carolina. It is sponsored by this biotechnology company. It is for HER2 overexpressing patients, and the way the process is done is the patient first undergoes GCSF mobilization, no, not GM-CSF, but GCSF mobilization, being, of course, the more commonly used and better tolerated drug, which again also increases the number of monocytes in the blood. The patients then undergo apheresis similarly to what one does with CAR T cells, and then we send the leukopak centrally for manufacturing. The cells, the monocytes undergo CD14 selection using a Clinimax-like device, and then those undergo a monocyte to macrophage differentiation, now using GM-CSF in vitro, followed by transduction with the clinical grade vector that we made. And then those are essentially CAR macrophages, a high percentage of CAR macrophages that, as I mentioned to you before, are highly activated. And those are cryopreserved, the cells are frozen, and then shipped back to the hospital for administration. The process is, the in vitro process, or ex vivo process, I should say, is 7 days long, and the yield is up to 5 billion CAR macrophages per patient. And this, as I mentioned, is done centrally. And so the phase 1 clinical trial that is now has now started at Penn and UNC is a phase 1 study, of course, and as usual, safety and feasibility, and then we test for efficacy using serial imaging as well as biopsies. There are 18 patients to be recruited. The first group is a standard sort of intra-patient dose escalation. The second group is going to be a particularly interesting one that will help us to answer one of the most important questions, I think, in tumor immunology, particularly in adoptive cellular therapy, which is one of trafficking. And this is one distinct advantage of using macrophages as opposed to T cells, which is that macrophages don't proliferate hardly at all. And so that allows you to label them to radio label them and really watch their trafficking over time. And this technology has been developed quite nicely by several groups. We have some interesting approaches to basically labeling the macrophages with zirconium-89 and then following the trafficking and persistence of those macrophages using what's called an explorer PET, of which I think there are very few in the country. One of them is at Penn, and has, as I understand, a very high signal-to-noise ratio, meaning that you can actually detect, along with the relatively long half-life of zirconium-89, you can detect persistence for up to 2 weeks in vivo. And then the patients undergo several biopsies in order to help us to ascertain the mechanism of response if we see a response, or the mechanism of resistance if we do not see a response. So in conclusion, the way I think of engineered macrophages, at least adoptively transferred engineered macrophages, is as a sort of Trojan horse phenomenon. I try to explain that, at least how I think of the fact that tumors generally recruit into themselves monocyte macrophages in order to perform crucial pro-tumor anti-immune cell functions. Those are TAMs or M2 macrophages, but that doesn't need to be the case. And in fact, what we are hypothesizing essentially is that we can use the very same mechanisms that the tumor microenvironment uses to recruit immunosuppressive macrophages in order to recruit non-immunosuppressive and indeed engineered anti-tumor CAR macrophages, essentially as a sort of Trojan horse. And so a lot of this work, all this work essentially was done in my lab by Mike Laczynski before he went to the company. I wanted to point out various funding sources that supported this work. Thanks for your attention.
Thank you very much, Dr. Gill, for that fascinating presentation. I'd like to invite everybody to place questions in the chat if they have questions. In the meantime, I will get started because I have a couple of questions. And so I think, one of the things that comes to mind seeing your presentation is that really this opens the door for lots of potential combinations of therapies as well. And so I was just wondering what your thoughts are on that. And in addition to combinations with some immunotherapies, it also opens the door to combinations with other agents, which could be targeted therapies, for example, as well. And I was just wondering whether this is something you've looked at or thought about.
Yes, well, we have looked at it. Actually, I don't think I showed you that. No, I did not show the data where we combine, or at least the company's labs now combine CAR macrophages with PD-1 inhibition, showing at least an additive effect. I didn't share that data, and perhaps I should have, but I think that makes sense. Whether it is PD-1 or PDL1 that functions on those macrophages, or on T cells, or on the macrophage T cell interaction, or on the tumor T cell interaction, I think that is a very, I would say, actually rational combination. A question that often comes up and that we've thought about, of course, is whether inhibition of the do not eat me signal with CD47 blockade makes sense. Perhaps, but I would actually put it below PD-1, because in many different experiments that we've done, it just doesn't appear that these macrophages are significantly inhibited by this SIRP alpha CD47 interaction. So that may not, although it seems rational, I think I would prioritize that below other so-called checkpoint blockade. You can also think of using these cells as delivery vehicles, and this is not necessarily work that we've done, but I think a really nice paper that I saw recently, I think it was in Cell, perhaps, where the macrophages were induced to secrete IL-12. And so additional ways of maybe breaking open the tumor microenvironment, releasing the immunosuppressive breaks in the tumor microenvironment. And in a way, perhaps that is more anatomically or regionally defined, because you get enrichment of these cells, as I mentioned, down the chemokine gradient in the relevant places. So yes, absolutely. Thanks for that question. There's so many different ways that this could go. Absolutely.
Thank you. And we have another question from Roger. Have you tried this method using antibodies against other antigens expressed on breast cancer or other cancers? So what are the other targets that you're thinking of?
Right. Yeah. Well, we concentrated initially on HER2 because of a variety of reasons. There aren't, as I sort of alluded to, I don't think there are amazing, great, specific, and 100% expressed, homogeneously expressed surface antigens in solid tumors. But amongst the list that one would be interested in, that I would say mesothelin would be pretty high, PSMA pretty high, and so those are the next ones, at least in my view, to try.
Thank you. And there's a question from Dr. Huliskoven. Would you like to ask it, Sue-Ann?
Sure, yeah. Thank you, Dr. Gill, for speaking. And I want to say, actually, I was asked to give a comment about your study and, the study you published last year and your clinical trial, or, from the clinical trial, from the company that you founded. At the beginning, when I was told about this concept, I was not very enthusiastic, because we all know macrophages are so mythical, right? They have so many different phenotypes, and it's just hard to manipulate them. But after looking at your study and looking at the results, I was really fascinated. What a great concept to utilize the cell type that naturally go to the tumor, right? And just manipulate it and make it, to the phenotype that can kill the cancer cells. So my question is really around the safety of this, right? So I did not see a lot of safety data from your clinical study. I'm just curious, how long these cells can persist in the mice after. I know you're still collecting samples from the patients, but just from the animal study, how long these cells can persist, and whether their phenotype change over time.
Yeah, thanks for that question. Thank you for your kind words. I would say that, well, the cells don't persist longer than about a month in the mice, for what that's worth, at least in the xenografted mice, for what that's worth, because it's a very, obviously, artificial model. And we will see how long they persist in humans after adoptive transfer. I don't expect them to persist for a long time, because macrophages don't. I mean, they don't like T cells do, but macrophages don't, generally speaking, and they certainly don't proliferate and certainly don't expand exponentially like T cells do. So I think the way I think of it now is that we should expect perhaps some of the work to be done by direct phagocytosis, but perhaps the real heavy lifting to be done by stimulation of additional immune effector cells like T cells. So when it comes to safety, well, you're right. There wasn't a lot of safety work done in the study, and there wasn't necessarily a lot that was requested by the FDA, because the targeting domain, as you know, does not cross-react with mouse tissue, so it really wasn't possible to study it in vivo. But what was possible and was done quite thoroughly, and I think it might be in the supplementary of the paper, is across a range of normal human cell panels, normal cells. We compared phagocytosis of those to phagocytosis of cancer cells, and the therapeutic window there, I very much believe, or at least based on the data that we have, the therapeutic window comes from the delta in expression of the target antigen. So that's why the clinical trial recruits patients with high HER2 expression, 3+, for example. And so, and that's where that occurs. And why do we feel like there may be, why do we expect there to be anti-tumor response, even though we know that HER2 is not uniformly expressed in all cancer cells? Again, that's because of the antigen presentation and stimulation of T cells. I think I saw briefly a question pop up in the chat about cytokine release syndrome. Yep. We never saw any sort of clinical evidence of cytokine release syndrome in our mice, immunocompetent or immunodeficient mice, and I don't necessarily expect to see it in the patients. It is true that CRS, we think CRS after CAR T cell infusion occurs predominantly by release of those cytokines, including IL-6, from myeloid cells such as macrophages upon stimulation by T cells as those T cells expand and proliferate. But that isn't the same here. And in fact, you may, I suppose I could do a back of the envelope calculation, but the number of cells that we're infusing into the patient, the number of activated macrophages we're infusing into the patient, is much lower than is a small percentage of the total load of macrophages in the body, where those macrophages are entirely susceptible to being stimulated by T cell mediated cytokines in the context of CAR T cell CRS. So I don't necessarily expect to see cytokine release syndrome here.
Thank you very much. Thank you for presenting today. And I will now turn it over to Dr. Huliskoven, who will introduce the final speaker of the day.
So before that, I actually have a last question for Dr. Gill. I just wonder whether HLH can ever be a concern, or whether you have thought about that. That's a condition that macrophage become overactivated, right? It's whether you think that might be a concern, or it's not really relevant for this situation.
Yeah, look, I don't expect it for the same reason. At least in the context of CAR T cells, it seems to be on a spectrum with severe cytokine release syndrome. So same comments that I think that go towards CRS go to HLH. HLH, other than CAR T cells, is mediated largely, of course, by histiocytes or macrophages, but predominantly in the setting of viral infections where there is insufficient T cell or NK cell shutting off of those macrophages. But regardless of what I think, we will see. And of course, the trial is designed to look at that as well. That is absolutely one of the criteria for safety.
Thank you.
Thank you.
Okay, so it is my pleasure to introduce our last speaker, Dr. Krishna Ganturu. So Krishna is a medical oncologist from Leahy Hospital and the Medical Center. She graduated from Dartmouth Medicine Residency and Yale Oncology, and she has been the director of survivorship at Leahy Hospital. So she is going to present as 2013, which is a clinical trial initiated from the Symptom Control and the Quality of Life Committee. It is immune checkpoint inhibitor toxicity. In short, ICHECKIT, a prospective observation study trying to categorize and catalog patients' toxicity profile after starting checkpoint inhibitors and trying to build up a model to predict the occurrence of IRAE. So at the last meeting, Dr. Ganturo actually already presented the concept, and now the protocol is approved. So she is going to provide an update.
Thank you very much, Dr. Hillis-Cohen and Dr. Politi for giving S2013 team a chance to present. So we have heard so far about fascinating work in the preclinical and clinical work in how to increase responses and survival with immunotherapies and related treatments. So let's ask the question of how we can make this longevity more enjoyable for our patients with the decreased toxicities and also by increasing their quality of life. I am not able to move the slides. Let me take control. Okay, thank you. There. So the simple question from this prospective trial is for all of you who's interested in immune checkpoint inhibitors or immunotherapy is, would you be interested in a risk prediction model for immune-related or IRAEs for patients receiving immune checkpoint inhibitors in solid tumors? So the primary objective of this study is to develop and also to validate a risk prediction model for non-hematological grade 3 or higher immune-related adverse events in patients receiving immune checkpoint inhibitors in solid tumors. So the secondary objectives and translational objectives of the study in the first 12 months since starting the immune checkpoint inhibitors, whether it is monotherapy with the PD-1, PDL1, or CTLA inhibitors, or combination of CTLA inhibitors with PD or PDL1 inhibitors, is to also look at the incidence of any grade non-hematological and grade 4 hematological IRAEs. We also plan to look at patient-reported quality of life and patient-reported symptom outcomes, aka PRO-CTCAE. We have seen a lot of data on reporting on the toxicity over time, like chronic low-grade toxicities, whether it is endocrinopathies or skin reactions, dermatological rashes, or other things that people are living as they're living longer now. So we plan to look at the toxicity over time, which includes the chronic grade 1 and grade 2 toxicities that our patients are living with. And also to look at the patterns of treatment of IRAEs and how this toxicity is resolving. So the translational objective of this study is to get a measurement of 11 different cytokine levels called cytox score. We plan to get blood or serum sample at prior to ICI and after first cycle to predict the development of IRAEs. And this is also going to give us a big, huge biorepository of archival tissue and serum for predictive and prognostic IRAEs risk for future studies. So if you don't remember any of these, the main thing you need to remember from the secondary objectives is the chronic toxicities and PROs evaluation in this study. So we kept this eligibility criteria very open. As I said, this is for immune checkpoint inhibitors, patients receiving in solid tumors as standard, which is FDA approved or NCCN 1 or 2A recommendations as monotherapy or combination with ICI, not with the chemo or bio or targeted therapy for solid tumors or on a clinical trial. The reason why we kept it clean is to have this homogeneous repository and background so that we can do the tool, prediction tool. So they can have prior ICI therapy, but must not have discontinued this because of the IRAEs. And they also should be recovered from the side effects of prior therapy per treating physician's discretion, which we outlined in the protocol, and able to complete PROs either in English, Spanish, or French. So if you're interested, what we included in the risk prediction model, these are the demographics and history, which includes age, sex, race, ECOG performance status, BMI, including autoimmune diseases and other treatment options that they had before. Along with that, we also included PROs, patient-reported outcomes, and labs, which are at baseline. So the accrual goal is 2,062 patients for this study. And the main things I want you to remember is the primary objective is trying to get a prediction score that we could use in everyday life in the clinical setting for our patients to see who would be the ones that would develop IRAEs. And if that is the case, whether we can use different type of treatment if it is available, or increase the surveillance for these toxicities so that we can decrease the toxicities for our patients. And also other things to consider is this is the real world setting that we use these medications for, where if people are not enrolled in other clinical trials or eligible for other clinical trials, these patients can be enrolled in this study. And this is very easy. There are only 5 time points where there are evaluations, which is at baseline, at week 4, 12, and 24 and 52. So those are the 5, and blood draws is at only 2 levels, including grade 3 toxicities if they develop any. So this is a low burden for our sites and for our research team. So this would be easy to enroll and also get accrual easily to answer this study. I would like to thank our study team, which are mentioned on this slide here, and happy to take any questions. This would be ready for enrollment in the next few months in early summer.
Thank you so much, Dr. Ganturo. I also want to invite Dr. Nicole Kudura. If you have your camera, you can turn on your camera to help answer questions. She's one of the key co-investigators of this trial and also an important member of the IO committee. So I want to repeat several key points here that Dr. Ganturo has outlined. So this is a large study, but the actual execution of the study is pretty simple, and there is any site can potentially enroll a lot of patients into this trial. And any solid tumor patients who are going to go through immunotherapy, and that includes anti-PD-1 by itself or combination strategies, can be enrolled into this trial, can be potential candidates. It's, I think that this is a nice study to enroll a lot of patients to NCI trials. And the execution of the trial is pretty simple. Nicole, do you want to add anything to that?
Yeah, I'm actually just typing. Thank you so much, Krishna. Wonderful job, and Soon and Kathy, and the whole SWOG team for making this happen. This was a wonderful team effort and is setting the stage for really toxicity research, bringing us to the next level to really have prospective real-world studies, as the highly selected trial patients do not give us the comorbid patient, the more vulnerable patient, where we expect potentially also greater toxicity. And there's some more details that we're hashing out on those additional risk factors as well. So this is a study for patients on standard of care, where we rarely have studies. So we hope that we're not competing really in any major way, and we appreciate you watching out for those patients and help us stratify these vulnerable patients for the future.
So thank you, Nicole. And Krishna, can you help to clarify, say if site want to open this trial, who should be the supervising, the PI of the trial, whether it's a specific tumor groups or a phase 1 team, what's the, just some logistic considerations, what team should be handling this trial?
So if other S2013 team is online who can answer that question, please feel free to jump in. So this is coming from SWOG, symptom control quality of life. My understanding is this can be generic, like it can be open in any solid tumor patients that would be getting immune checkpoint inhibitor. So, and also it would count for your SWOG or NCARP also accrual too. So if that answers your question, Steven.
Yeah, so the question is what kind of a team should handle this trial at a specific site? But I guess, I understand your answer is whoever is interested, right? And because it's open to all solid tumors.
Yeah, it would be because we're mainly, they're filling up the patient-reported outcomes, which can be paper and also on the tablet too. So all I think we need is a usual research team, which includes the CRA and nursing. It's also mainly taking the CTCAE grading. So we have all the forms and everything. So I don't think we need any special experience for this study.
Very exciting. Is this the largest trial that collecting IRE reports?
To date. And so this would be a huge biorepository bank that can be of anyone's IO in the IO world's dream for any future studies.
Yeah, that's great. And the protocol is approved, and I encourage everybody to consider opening this trial at your site. So Katie, do you want to give a concluding remarks before we close the meeting?
Thank you very much, everyone.
Sure.
Thank you.
Thank you very much for that presentation. I think I'd really like to thank everybody for speaking today. And I think we covered a lot of ground from talking about the IMatch clinical trial and that's rollout and development, to talking about immunotherapy and resistance and overcoming resistance and new targets, to CAR macrophages and to toxicities to immunotherapies. So we've really covered a whole spectrum of aspects of immunotherapy research, and thank you very much for everybody for participating. And please join us, reach out to us if you'd like to join us for our monthly meetings. The next one is on May 7th, Friday, May 7th at 2:30 p.m. Central. And Siwen, I'll turn it over to you for any final comments.
Yeah, I just want to welcome any SWOG members to join our committee. We're a young committee and we're still defining and redefining our mission. So it will be great to have your support in this process. And thank you so much for attending this meeting. Hopefully next time we'll be meeting in person. Have a the rest of the afternoon.
Thank you very much. Bye-bye. Bye-bye.
Take care.